Okay. Assalamualaikum warahmatullahi wabarakatuh. Okay, let me introduce myself. My name is Khairunnisa Raudhatul Jannah Syukra. You can call me Nisa. I'm a medical student in Bengkulu University, and today I will tell you my experimental about effect of yogurt fermented with Lactobacillus delbrueckii SSP bulcaricus OLL1073R1 on the IgA flow rate of saliva in elderly persons residing in a nursing home, a before after non-randomized intervention study. Let's we see the first slide. Okay, first introduction first, right? This is an oral cavity. Yeah, oral cavity represents the beginning of the aerodigestive tract mucous membranes and is one of the most frequently exposed sites to foreign antigens, as it constantly encounters foodborne, waterborne, and airborne antigens and other environmental insults. According, immune surveillance in the oral mucus is highly important for both local and systemic immunity. The oral immune system is part of an extensive and specialized compartmentalized mucosa associated lymphoid tissue. The oral cavity is protected by saliva, which is a biofluid produced mainly by three pairs of Meyer's saliva glands, the submandibular, parotid, and sublingual glands, along with secretion from many minor submucosal salivary glands. Saliva plays an essential in controlling oral infection and in protecting the teeth from caries by diluting and eliminating sugars and other substances and via its antimicrobial actions. Saliva contains several antimicrobial agents, with the major and most important antibody being immunoglobulin A or IgA. The monometric fraction of salivary IgA is generally small, and most IgA in the saliva exists as secretory IgA, a dimer. IgA plays a critical role in the defense of mucous membranes against foreign antigens and pathogens, directly neutralize the activity of pathogens and their toxins, and contributes to the creation and maintenance of mucosal homeostasis. A lack of nonspecific IgA at the mucosal surface or the inability to produce specific IgA can lead to an increased risk of infection. Recently, uh, there is several studies have reported that certain strains of lactobacillus and their products can modulate the immune response and change the intestinal bacterial flora. For example, intake of Lactobacillus acidophilus and Lactobacillus paracasei was found to result in increased numbers of IgA. And then interleukin and interferon gamma producing cells in the small intestine of mice. Lactobacillus delbrueckii bulgaricus increased the number of Clostridium perfringers and decreased the number of Bacteroides. And many strain order that can increase the IgA level. Although many reports have indicated the various immunomodulatory effect resulting from intake or Lactobacillus SSP and their products, there are currently few studies on the effect of on the IgA level in human saliva. So, in this study, we hypothesized that Lactobacillus bulgaricus OLL1073R1, which has already been shown to reduce the risk of colds, may increase the salivary IgA level. To address this hypothesis, we examine the alteration in the salivary IgA levels of elderly persons residing in a nursing home after daily intake of yogurt fermented with Lactobacillus bulgaricus OLL1073R1. So, The participant of our experiment uh, intervention before after non-randomized controlled study conducted in a population of elderly volunteers residing in a single elderly nursing home. The study was conducted in a stage from October to December 2013. Individuals taking antibiotics or morphine with gastric fistulate who disliked yogurt or who were hospitalized and died during the study period were excluded from the study. So, as a result, the study finally enrolled 37 participants, 7 men and 30 women, with a mean age of 82.7 years. Range is 64 until 101 years. Um, the level of care required was scored as 1, 2, 3, 4, and 5 in 2, 8, 7, 14, and 6 participants respectively. And then the dementia level was scored as 1, 2A, 2B, 3A, 3B, 4, and M in 2, 3, 5, 10, 9, 6, and 2 subjects, respectively. And then, in terms of the physical activities level, daily living, and the dementia level, none of the participants were able to live at home. None of the participants were smokers or drinkers. Data on the participants are age, sex, height, body, and body weight. Next, ethical consideration. This study was approved by Ethics Committee of Kanagawa Dental University in 2013 and conformed to the principle outlined in the Declaration of Helsinki of 1975 as the revised in 2008. On implementation, all participants and their families were given written and verbal explanation regarding the purposes and methods of the study, any potential risk, potential benefits, protection of personal information, and freedom of consent and withdrawal. Subsequently, all participants or their families signed the informed consent form. So, the experiment. First, the participant ingested 112 grams of yogurt fermented with Lactobacillus bulgaricus OLL1073R1 each day in the morning. That is 10 a.m. to 10:30 a.m. for 12 weeks. The care staff wrote down the yogurt intake of the participant each day. All participants lived in the same elderly nursing home, hence their dietary constituents, meal times, breakfast at 8 a.m., hour rising at 7 a.m., and bedtime at 9 a.m. were basically the same. The daily calorie intake of the participants was approximately 1,600 kilocalorie. The room temperature of the nursing home was kept as 22 Celsius degree. Saliva samples were obtained in the morning at 9:30 to 10:30 a.m. before 0 weeks and after 4, 8, and 12 weeks of yogurt intake. And then the yogurt used in the study was provided by Meiji Co. Ltd., Tokyo, Japan. The yogurt was the same as that of the marketing product of the company. The yogurt was prepared using two lactic acid bacterial strains, namely Lactobacillus bulgaricus OLL1073R1 and Streptococcus thermophilus OLS3059. These strains were originally isolated from traditional Bulgarian yogurt. The cell count of Lactobacillus bulgaricus and Streptococcus thermophilus in the yogurt were standardized by Meiji Co. Ltd. Refrigerated yogurt were delivered to residents of each subject once a week and stored in the refrigerator until intake. Next, measurement of IgA concentration. The concentration of IgA in the saliva was quantified by enzyme-linked immunosorbent assay or ELISA using a human IgA quantitation kit. microtheater 96, while plates were coated with gold anti-human IgA diluted 1 and 100 with a coating buffer for 1 hour at room temperature. Washed 5 times with wash solution and blocked with blocking solution. And then for 30 minutes at room temperature. After washing with wash solution was described above, the samples and human IgA standards were added to each well for 1 hour at room temperature. After washing, horseradish peroxidase conjugated gold anti-human IgA detection antibody was added to each well for 1 hour at room temperature. The reaction was developed after the addition tetramethylbenzoic substrate in the dough at room temperature for 15 minutes and stopped, after which the absorbance was measured at 450 nanometer using an automated microplate reader. And then next to statistical analysis. All statistical analyses were performed using GMP version 12 software, and the results are expressed as the mean and standard error, or SE. The statistical analysis for difference in the body weight and body mass index, or BMI, before and after 12 weeks of yogurt intake were performed using Welch t-test. Statistical analysis for difference in the flow rate of saliva, concentration of IgA in the saliva, and IgA flow rate were performed using factorial two-way analysis of variance, or ANOVA, including two main effects and one interaction effect. Next, so we have some result. First, there were no significant difference in the body weight or BMI between before and after 12 weeks of yogurt intake. And then the second one, the IgA flow rates of the saliva at 8 and 12 weeks were significantly higher than that at week 0. And then both the flow rate of saliva and the IgA flow rate of saliva were affected by participants' body weight. Then, the IgA flow rate of saliva was calculated by multiplying the absolute IgA concentration by the flow rate of saliva. Therefore, the flow rate of saliva influenced the IgA flow rate of saliva. In the present study, the participants' body weight was low. The flow rate of their saliva tended to be high. Previous studies have reported that the flow rate of saliva in elderly persons is decreased as a result of atrophy and decreased acinar cells in the salivary glands, partly resulting from an increase in adipose tissue of the salivary glands. In the present study, when the body weight was high, the flow rate of saliva tended to be low, suggesting that the adipose tissue in the salivary glands of participants with a high body weight might be more increased compared to in participants with a low or normal body weight. In cases of very low body weight, the flow rate of saliva was also relatively low. In accordance with this finding, it has been reported that the unstimulated flow rate of saliva in elderly people with malnutrition was low. Those elderly people with either very low or high body weight are more likely to have decreased flow rate of saliva and IgA flow rate of saliva. Of note, underweight status was found to be a risk factor to higher mortality caused by infection for elderly nursing home population in one previous study. And in our previous research, the IgA flow rate of saliva significantly correlated with the body weight of the study participants. In addition, one-way ANOVA showed that the height of the participant didn't affect the flow rate of saliva, concentration of IgA in saliva, and IgA flow rate of saliva. And then consideration from our research this research is the first one. Our study didn't include a yogurt non-intake group as a control population. Hence, we couldn't clarify the effect size of intake of yogurt fermented with Lactobacillus bulgaricus OLL1073R1 on salivary IgA in elderly persons. Second, we couldn't rule out the existence of confounding factors, including the effects of seasonal change on salivary IgA. However, Wagner reported that no seasonal change was observed in the salivary IgA level in children, and we therefore consider that the salivary IgA level in the elderly person of the present study was not highly affected by seasonal change. And then the last one, the yogurt used in the present study was the same as that of the marketing product of the company. Thus, we were not able to measure the number of bacteria included in the yogurt. So, however, the cell count of Lactobacillus bulgaricus and Streptococcus thermophilus in the yogurt were standardized by Mexico Ltd. So, finally, the conclusion from the research as continuous intake of yogurt fermented with Lactobacillus bulgaricus increased the IgA flow rate and concentration of IgA in saliva of elderly people residing in a nursing home. Moreover, we found that the flow rate and IgA flow rate of saliva were affected by the body weight of the participant. Taken together, our results suggest that possibility that continuous intake of yogurt fermented with Lactobacillus bulgaricus may represent a suitable strategy for improvement of mucosal immune function in elderly people who tend to have weakened immune system. So, I think that's my presentation from my result of my sources. Thanks for your attention. I hope this research uh, can be used in the future. So, thank you very much for your attention and Assalamualaikum warahmatullahi wabarakatuh.